And I would point out that today our understanding of brain function, our understanding of what is the source of rage, what is the source of depression or anxiety, is still at an incredibly rudimentary level. And studies being performed on this campus now and elsewhere, in this case, studies that use both invasive and non-invasive procedures in both humans and non-human primates, are being used to try to identify what is the function of the brain, how does it function. And interestingly enough, the rhesus and the human tend to show exceptionally similar responses in this area. So here's one organ where we're still at the very basic level, and according to the criteria that Ray provides, this then also is an appropriate subject for animal modeling. Here's a trickier case, Harry Harlow. Trickier because of all of the ethical aspects associated with the work that he performed. I suspect there will be rather broad agreement in this audience that the studies that he performed should not be performed now. And perhaps not just because that would needlessly repeat something that's already been done, but because all of us perhaps might feel that those studies are inappropriate as ways to treat animals. And I think there will be a number of people that consider that those studies never should have been done at all, but they were performed. So let's look at them. Let's look at this difficult case and see whether or not that large series of studies was predictive. So the general question he was asking was, what is the connection between human development, affection, and love? And the paradigm that existed at that time in the 50s and 60s was that affection created needy and demanding offspring, and had the likelihood of spreading infectious disease if touch was included as part of it. And using rhesus monkeys, he performed a large series of experiments with cloth mother, wire mother, abusive mother. So these were studies where, for example, an electric current was put through this substitute mother, this surrogate mother, to shock a child that was clinging to it. And that's where the ethical issues come in. But again, we're talking about prediction today. He also used methods of social isolation, also filled with some ethical implications. And his findings were that touch, facial expressions, particularly of the mother, that affectionate interactions, expressions of love, these were all fundamental to healthy mental and emotional and physical development. Again, a dramatic break from what was the dominant paradigm at that time. Deborah Blum, who is on campus here, wrote a book called Love at Gould Park, which describes Harry Harlow and his work. And I would like to read how she finished her book. So let us remember the best of Harry's contributions as well as the worst. Let us not slip backwards ever into believing that we are not necessary to each other's health and happiness. Now she's talking about humans here, us, each other. You don't have to like the way Harry found his answers. Almost no one could admire every choice he made. But neither should we pretend that he did anything less than arrive at some fundamental truth. Our challenge is not to squander it. So there's a lot in there. There's a lot about how we as humans should deal with each other and treat each other, about how we need each other. And all of this was based on Harry Harlow's work with non-human primates, with the rhesus. This series of studies was exceptionally predictive for this aspect of human biology, human psychology. And it dramatically changed our understanding of human relationships. So now I'll move to something a bit more like what you were probably expecting me to talk about. Michael Gould and breast cancer susceptibility. Michael is a faculty member on this campus and has done for a long time a large series of studies using the rat as a model to study mammary cancer. Listed at the top is the title of the article that I'm referring to. I'm not going to read it. You can read it yourselves. As I said, he's using rat models of chemically induced mammary cancer. And by using 2 strains of rats, Michael and his colleagues identified 4 regions on different chromosomes in those rats that influence rat mammary cancer susceptibility. Furthermore, and I think this is the most important thing, they then examined the human equivalent of the first chromosomal region. They still have 3 more to look at. They've looked at this first using human population-based studies. The type of study is mentioned there. So looking in people. And they found the same susceptibility modifier. So I'm going to give you a couple of slides that any scientist is required to present in any presentation, and that involves some jargon. It was one of the things that I had to sign that I would do when I got tenure. So everybody has their own particular kind of nomenclature, their own particular kind of record keeping. Bookkeepers use spreadsheets. Electricians use wiring diagrams. Frustrated parents on Christmas Eve use poorly written assembly instructions. This is one of the ways that geneticists present the information that they've learned. And it's actually relatively straightforward. And I don't have a pointer, so I'm going to step away and try to talk loudly enough so that you can hear me. And if you can't in the back, please let me know. So on the bottom is just a diagram of a portion of the rat chromosome 5, which has been generated via the genome mapping studies that have been done that include multiple species, including humans. These various lines represent the genetic composition of some of the rats that Dr. Gould and his colleagues used in their studies. And up at the top, MCS5A1, MCS5A2, those are 2 regions that are part of the MCS5A locus that they identified. And MCS stands for mammary carcinogen sensitivity. Okay, so this is in the rat. What about in the human? I'm going to show you a very similar kind of a map now. Along the bottom is the corresponding region of human DNA. It happens to be on the human chromosome 9. And at the top show the same 2 regions of susceptibility. But I think the most important thing that this slide illustrates is that, and now remember, this was identified in the rat. On the left, for the MCS5A1, the dark portion on the left, 6% of women in this large study, and the study was a series from Wisconsin and from the United Kingdom, 6% of women carried genetic makeup at that MCS5A1 that gave them a 19% increased breast cancer risk. Looking at the other half, 22% of women in this population had a genetic composition at that area that gave them a 14% reduced breast cancer risk. So what does this really tell us? In this example, what is the rat telling us about humans? So in summary, the genetic studies by Dr. Gould and his colleagues identified cancer susceptibility DNA that predicted the same areas of DNA with the same effects in humans. And interestingly, human studies themselves had not been able to identify these areas. The reason for that is the human population is very diverse. The background noise is very high, whereas rat strains are inbred. So that gives us an advantage. It's one more way that these animals are different than humans, but a scientist can use that fact, can use his or her understanding of that difference and turn it into something, if it's used appropriately, that is predictive of a biological phenomenon in humans. In this case, predictive of human susceptibility to breast cancer. And they still have these 4 other loci to look at, these 3 others to look at. So stay tuned. So what do we know? Scientists recognize both the similarities and the differences between species and between individuals. That's kind of a duh. We don't think that there's a one-to-one correspondence between a mouse and a human. We don't think that there's a one-to-one correspondence between a non-human primate and a human primate. And I would agree with Ray that there's not even a one-to-one correspondence between any of us in the audience. But using this information about similarities and differences that we've accumulated over a lot of years, we can use animals in the appropriate ways to make accurate predictions about humans. And animal models have contributed immensely to our understanding of human biology and to our understanding of human disease. And the reason is because rather than emphasizing the differences and giving up, scientists identify and take advantage of the similarities and use those as ways of learning, of taking information from one species into another species. What's the future? Biomedical scientists will continue to develop and employ advanced methods to answer questions. So science is an incredibly rapidly evolving field in terms of the methodologies that are available. Ray talks about some of these on his website, ways of performing tests in humans. And in fact, many of those same things are being adapted to use in animals. So scientists don't simply perform the same studies over and over again. Not good scientists. Rather, they change their approaches and change their methods as the information that we do understand changes. And as we develop a greater understanding of animal and human biology and disease, we become better at choosing the right animal model. And by right animal model, I mean the animal model that is most predictive for a certain type of study. And finally, more studies will address translational medicine. And that is the application of information from animals to humans, trying to take what we've learned in animals and specifically use that and transform that into a pattern that has meaning in the human population. Sorry, I got distracted by that phone call, and I was just thinking that, thank God mine is turned off. My daughter taped a song for my ring, and I don't know how to change it, and it's very embarrassing. Anyway, and then also more studies will employ human subjects, and many studies right now are doing that. In fact, here is an example. So these are some data from UW-Madison, as reported to the United States Department of Agriculture. There are certain covered species. There are certain species that the USDA regulates whose numbers we have to report each year. Rats and mice and birds aren't included. You'd have to add 60,000 rats and mice to these numbers. But you can see the numbers of dogs, cats, guinea pigs. And boy, I don't know why experimental subjects are called guinea pigs anymore, because certainly there's not very many experimental subjects that are guinea pigs. Hamsters, rabbits, non-human primates, those all add up to about 4,000. And in fact, during that time, about 4,000 humans were enrolled in clinical trials here. So we on this campus are not ignoring humans as a subject for human medicine. Rather, we're combining animal data with human data, sometimes in the same laboratories, and using that information as a way to gain an understanding, to gain predictability about human disease and animal disease. And finally, I want to make a point that can get lost in this argument. And here's where I kind of want to take a step and just cross the room. And not so much worry about dividing everything by half, by half, by half. Let's take a look at clinical medicine. So if animals are to be predictive for humans, you'd expect humans to be predictive for animals. And in fact, it does go both ways. And on this campus, we have a chance to see this. I work in a veterinary school. There's a large medical school on this campus. And this slide is the result of a 5-minute conversation I had with one of my veterinary colleagues, where I asked, what are common clinical treatments for conditions? Let's pick dogs, one of the probably the most common companion animal species. Dogs or cats, can't remember which is most common. I'm sure most of you know.